Today we talk about proteomics in real-world application. And now I welcome Sasha. So Sasha is assistant professor at the Women's Brigham Hospital in Boston, and she's really an advocate for LC-MS-based proteomics among all the clinicians and scientists worldwide, so that's really, really great. And today she will talk about a practical approach to plasma proteomics, focusing on data quality and, which is very, very important. So, Sasha, welcome to the stage.
Thank you, Garin, and thank you, everyone, for attending today. Beforehand, I apologize. Some of the slides didn't transfer very well. They're a bit blurry, but most of the work I present today has just been published. So being practical about plasma proteomics, emphasizing quality over quantity. When a new technology emerges and everyone gets excited, sometimes it becomes very challenging to be practical when quantity is often the benchmark for success. And just a bit of a background in the lab, we do in fact have a proteomics center within our center. As Garwin said, we have about 40 collaborations around the world that are greatly facilitated by the proteomics workflow. We have a station here where we keep track of all the kits that come in and out. And our very first publication, I believe, was around 2019. And about 80% of our publications since have been enabled by the proteomics suite. So just quite recently, last summer, we finally had the courage to venture into the biological fluids. And again, with the support of the various platforms provided by proteomics. Today I'm going to talk about the Enrichment Plus that's no longer available and just give you a preview of some of the work we've been doing with the P2. So I don't know how many of you can relate to this. So we, about a year ago, we entered the plasma proteomic field, pretty proud about our 1,000 proteins that we presented last year, only to be dejected by all the other posters that we saw that we're getting 5,000, 6,000, 7,000 proteins. And it caused a lot of soul searching for us. So we went back to the lab. We worked, went to the proteomics guys and said, hey, what's going on? Is it us? Is it them? Is it you? What is it? And more or less, I'm summarizing a study that just got published in JPR a couple of months ago, but I also want to highlight a really nice study that follow-up, I think, complements our work well from the Marcus Benchoff group, where from the title, it looks a little discouraging that pre-analytical biases, how variations in standard operating procedures may impact your data. But actually, if you read between the lines, it's very positive messaging that we're in a very good space right now with nanoparticle-based plasma proteomics. And if there's one thing that we can take home today is this particular graph. For a very long time, the abundance distribution of proteins in neat plasma have been a challenge for us. And primarily this right skew where very few proteins dominate plasma and we can't see beyond them. And the goal of us in the mass spec community is to compress that curve and bring more of those proteins into the dynamic range. But what we have to remember is that we cannot do this without compromising the proteome. So before we get into the mass spec, as I said, the lab did some soul searching. So we went back in history a little bit and tried to understand how standard operating procedures today were affected by the genesis of the plasma protocol. So what is plasma? A lot of people have been asking this question given recent advances and what mass spectrometry has taught us. So if you go back, plasma originally meant to form or mold something. Over time, it became a word to describe the modulation of your voice or to disguise yourself. And eventually, in early English, it became to describe a vessel or a pot. So we probably know plasma as ionized material or gas that was defined by Irving Langmuir. But for today, I don't know how many of you know this, Jan Purkinje of the Purkinje fibers or Purkinje cells in your brain is credited for defining plasma as the clear liquid remaining after blood has been cleared of its various corpuscle components. So what are corpuscles? Of course, the platelets, red blood cells, and white blood cells. So is plasma simply the leftovers? Or should it just be the leftovers? So to understand again how plasma has come about today, we have to go back in history. So to understand plasma, you have to know about the history of blood transfusion protocols. For millennia, even as late as the 1600s, whole blood was used in transfusion protocol, transfusion therapy, animal to human, human to human, and cadaver to human, with the aim to cure disease and even to rejuvenate people. But by the mid-1800s, it was clear why this wasn't so successful, because actually plasma contains many toxins. And the very first toxin that was described was fibrin itself, thereby generating anticoagulating reagents, namely heparin and eventually sodium citrate came about. And now we use EDTA. And then in the early 1900s, blood cells were also deemed toxin because of incompatibility with the different blood types, and there was born the blood grouping tests. So eventually, by the mid-1900s, transfusion fluids were available, including blood, but then plasma and serum became very popular, and stockpiles of it was made, and I don't know if anyone knows why. World War II. It was much easier to transport plasma to Europe in either a desiccated or dried form, where it could be resuspended in water, to treat wounded soldiers on the field. And here's a good book I recommend reading, where plasma or serum in its liquid state is the simplest and most economical means of preservation. So in other words, plasma was not generated or formed for the purpose of biomarker discovery. It was used as a therapeutic agent at that time. And here I list some pioneers of the transfusion biobanking protocols. And all the protocols that we know today were due to Charles Drew, who's the first medical director of the Red Cross. So we thank him for that. One more important person we need to discuss is Edwin Cohn. I think he's the first plasma proteomics person. And he, after doing experiments in plasma where he actually used electrophoresis, this is not a chromatogram. This is an electrophoresis of the main blood plasma components. Some of you may know them as the alpha, beta, and gamma globulins, which are actually not one protein, but many proteins that we've all come to know as the top 50 or so that suppress all the low abundant proteins that we wish to see. But he called plasma at that time to consider plasma as a system with many protein components and to explore, in light of recent advances, separation technology, chemical nature, and the physiological functions of the protein components, meaning the proteins themselves, if isolated, could also be therapeutic agents. But now we turn to plasma not for necessarily its transfusion potential, but for its biomarker discovery. And a very simple PubMed search shows that there are over 100,000 results in plasma for a biomarker, and it also covers metabolites and other molecules. So that brings me to this. So what is a standard operating procedure for plasma? Can we all agree on it? ChatGPT gave me this. Louisa Weiss, who is our expert in-house biofluid, agrees. And here I've just highlighted some of the components I think we all agree on. And I'll get back to that in a minute. And in terms of the proteomics field, it's, people have been interrogating how variations in plasma prep can impact the plasma proteome. But none of these studies have benefited from the deep proteome profiling technologies that we have today. So they would have had to have used dozens of samples or dozens of methods in order to see the differences owing to variations in standard operating procedures. So now it brings me back to this. We went back with our 1,000 proteins and wanted to know why we weren't doing as well as everybody else. So we did a very simple experiment. We said, let's order plasma from the five most commonly used sources in the United States of America, because presumably everyone at ASMS was using one of the five. We tried our best to kind of balance the demographics a bit. We pooled them, and then we had the Enrich Plus at the time, and we compared to the standard IST-BCT kit. Thanks to a collaboration with Bruker, we were able to have access to their timsTOF. In our lab, we have one of our instruments is Explorus, and you can kind of see we did everything. We used various software and various methods to collect the data. And so here were some of the criteria based on what was deemed standard operating procedure. We requested plasma with limited freeze-thaw cycle. We requested platelet poor preparations. We focused mostly on EDTA, and we had one citrate-based, one company that exclusively used citrate, so we took it in. We ordered it, and we also said, please ship it frozen. But interestingly, source 4 was actually delivered to us at room temperature. So we said, you know what, let's try. Instead of sending it back, let's see what happens. And then source 1 was repeated or done by hand by 3 independent authors on the paper. So when you look at the chromatograms, at first glance, they're very reproducible. They look consistent after preparation of the 5 sources. But when you start to look closely, it's more noticeable, I think, on the Explorers. There are differences. And that was one sign already we knew something was going to be interesting. So so I'm going to compare all 5 sources, but just to kind of orient you a little bit, the first 2 plots pertain to the Explorers, in which we have Chimeris and Diane. And then to kind of compare the 2 instruments, we also have Diane and Spectronaut. Diane for the timsTOF, and we also analyze the data with Spectronaut. And not surprisingly, you see a large difference in terms of the protein yield or the proteome, a number of proteins, but that's really owing to the different platforms of the instruments. We accepted this. And of course, there are differences based on the software that you use. But now you have to look across the different sources. And again, if you were to rely on the timsTOF alone for this comparison, nothing obvious may jump out, but the Explorers is more revealing. Namely, we have about a 2-fold increase in the proteome yield. And for plasma proteomics, that's huge. Maybe 5–10% you would expect when you have differences, but a 2-fold was very striking to us. So we also, again, compared. Now I'm just focusing on Chimeris on the Explorers data. The IST-BCT, that's the presumed neat plasma. We haven't perturbed anything in the proteome. We take it as the ground truth of the proteome. And sorry about this blurriness, but more or less you get a 2 to 3-fold or even higher, more yield of the plasma, which makes sense. But the PCA plot is actually very also revealing. I see 4 of the 5 vendors in the neat plasma cluster very closely, which is expected. But one of them seems to be moving away from there. And again, that's telling that there's something different about source 5 than any other of the sources from the neat plasma, meaning that neat plasma had already been perturbed in some way before entering the enrichment kit. Another clue that something else was wrong. So remember, I have the theoretical ideal situation where you're going to compress your dynamic range into a more narrow window. We see the peak in the neat plasma for all of them is distinct from the IST enrichment. But again, the source number 5 overlap very well, indicating that even the neat plasma has a more suitable compressed curve even before the enrichment protocol. So we looked at the data in many ways, but it was very obvious that we had 2 types of plasma proteomes. Remember, the same workflow, but 2 very different distributions. And we could group sources 1 and 2 as group 1 and sources 3 to 5 as group 2. And after looking at the data in different ways, no surprises. The proteins that define the 2 groups are primarily platelets, highly enriched in groups 3 to 5, whereas we have the canonical apolipoproteins and lipid-associated proteins that we often see in neat plasma defining groups 1 and 2. We also had ELISAs of the neat plasma supporting that the platelets were overrepresented in 3 of the sources. And source 5 platelets, ELISA values were actually beyond or out of range of the assay. So there were a lot of platelets in that sample. So once we realized, okay, it's not us, it's not you, it's them, we went back and we contacted all 5 vendors and we said, hey, This is what we requested. I think something else was going on in your samples. So sources 1 and 2 actually followed what we considered conventional standard operating procedures. But sources 3 to 5 were quite different. Storing blood on ice for prolonged periods of time, platelets don't like that. They will lyse. And so that kind of made sense that's why we had the high platelet counts. Source 5 or vendor 5 never got back to us. So we have no idea what happened behind the scenes. But what was interesting about source 5, something that we did not anticipate, was apheresis. And apheresis can be used for the purpose of blood donation for research, but also as a therapy to filter the different corpuscles, components of blood, to remove from a patient or to introduce a specific component back to the patient. And this company would have kept the plasma after the apheresis protocol. Be careful. ACDA is the only FDA-approved anticoagulant for apheresis. So if you need to order ACDA as an anticoagulant for platelet research, make sure the blood was not apheresed, because you could have had premature. The downstream steps could have still made the leftover platelets susceptible to activation and lysis. So from this part, some perspectives and positives from this. So if not for the bead enrichment, we would have not known about the variations in the standard operating procedures, because the neat plasma preps did not reveal that. ACDA, as I mentioned, is an anticoagulant, may indicate apheresis when you're ordering. Apheresed blood itself, depending on the protocol, may also deplete lipoproteins. So removing the lipoproteins is like doing a depletion step, and that is also why we may have been able to see deeper into the proteome. And alterations to plasma will impact specific laboratory assays, not just for proteomics, but if you're doing extracellular vesicle research or any other kind of platelet research, for example. And we actually think the apheresed blood is good because it could be a potential peptide library for actual samples. So there's been a lot of, I would say, negative stigma about platelets. Nobody wants to see platelets in plasma preps. But that's not always a bad thing. You have to be careful, obviously, to avoid that, their premature lysis. But there are many disease states, individuals whose baseline platelet activation is active. So if you don't know, platelets are from megakaryocytes, and they circulate in blood, and when they sense damage, they release a lot of beneficial factors into blood. So for example, here's an example of an aortic aneurysm in which platelets have been activated, but not just that, also leukocytes, white blood cells, extracellular traps by neutrophils. So a lot of cellular debris will be present in plasma. So the presence of these proteins in your plasma preps, assuming it's a disease state, does not necessarily mean poor sample preparation. We, in fact, expect to see that in our samples. So for us, the more realistic situation is because we do have now collaborations in South America and Africa where plasma is being transported to us. We cannot expect that standard operating procedures are going to be analogous to North America. Room temperature in sub-Saharan Africa is not room temperature in North America. In our context, because we study cardiovascular disease, almost all of our patients are dyslipidemic. They are going to have high levels of lipoproteins and fats in their blood. And so we don't expect for these patients as high yields as, let's say, someone with cancer or some other indication, or someone that has their lipid profiles under control. So just before I finish, now that we have a hold, we think, on quality, we want to get back to quantity. And because the Explorer 480 is kind of our workhorse in the lab, we thought, could we squeeze a little bit of more protein IDs out of our samples, the exact same samples? And the short answer is yes. So how did we do that? If you slow down the duty cycle, on the explorers for a very long time and increase the ion fill time, you can actually improve the number of IDs. And here's a plot that shows it. Sorry you don't see that. You can do that by increasing the resolution. It slows down the instrument. You have more signal to noise of your MS2, and you will get higher quality MS2 spectra. But there is a trade-off. As you increase the fill time, one, you slow down the instrument to very slow speeds, and you get fewer points per peak, okay? That's going to compromise, presumably, quantification, but you will get a boost in your IDs. And here we go. We show that inject, this is using the P2 kit, the same sample, but just changing the Orbitrap resolution, increasing signal to noise. You will get a higher number of proteins. You're not going to match an Astral. You're not going to match a timsTOF, but maybe if you're a lab, that's good enough. How is quantification impacted? We're now looking at that. In some cases, it's not. In other cases, it is. But I think it's good to know that we can exploit the versatility of these instruments to see more into the plasma proteome. So to end, what is plasma? This is my personal opinion based on my historical adventure. So I think it's a biofluid whose initial isolation protocols were optimized for the preservation of its therapeutic potential. But in light of recent advances, it can be optimized for the preservation of non-canonical corpuscles and signatures that mark early, mid, and late stage disease. And with that, I'd like to thank you for your attendance. And if you want to see more about our poster or ask any questions about some of the ways we operate the Orbitrap to get higher IDs, please visit Taku at his poster tomorrow. And we have another poster by Mahmoud where we look at DIA data to infer mouse apolipoprotein proteoforms in our data. With that, thank you.